Terrific.
You guys could duke it out? Terrific. Excellent. All right. So I was looking at my calendar today, and I saw that we have an exam a week from yesterday. I know that'll make all of you very happy. But at least there'll be good weather this weekend for studying. It's supposed to be about 85 degrees. You can sit outside and study. Wouldn't that be fun? No. Okay. Today I'm going to say a lot about transcription and get you through that. Last time when I finished up, I had finished up talking about termination. I talked about factor-independent and factor-dependent termination. And I'll remind you that the factor-dependent termination was the one that uses the little row protein, that's the little circle, and scoots up the rope, as it were, and catches the polymerase and basically lifts it off the DNA and causes everything to fall apart, at which point the RNA that's been made falls off. I should also say that all of the types of transcription that I'm talking about here, and the only types of transcription that we will talk about in terms of the synthesis of RNA, all involves messenger RNA. Okay? So transcription also describes the synthesis of ribosomal RNA and, I can't spit it out, the transfer RNA. All right? But those transcriptional controls I'm not going to talk about. Okay? And you won't be responsible for it. All right. Well, what I want to say now is talking a little bit about regulation, because regulation of transcription is really, really important because, as I noted yesterday, the more transcripts, that is, the more messenger RNAs that a cell makes for a given protein, the more of that individual protein that they make. So one of the controls that cells have over their destiny, as it were, is controlling how much protein they make. So cells have in them typically up to, in the case of a bacterial cell, a few thousand proteins that they can make. They don't make them all in equal quantities, and they don't need them all in equal quantities. So adjusting the quantities of the protein that they're going to make is really critical for a cell. All right? Because if they make too much of a protein, then at the very least, they're going to waste energy. And at the very worst, they might make a protein that catalyzes a reaction that makes too much of something that would cause a problem. Okay? So they really need to balance how much that they make. And there are quite a few controls that cells have over determining how much of a given protein that they make. So controlling transcription of the gene for the protein is one of the things that they have some control over. So we want to spend a little bit of time thinking about that. Yesterday I talked a little bit about the sigma factor. And the sigma factor, I said, was a protein that bound to the RNA polymerase, and it helped it to decide what's a good promoter. Where's a good place to stop and start making a messenger RNA? That's what the sigma factor does in a prokaryotic cell, in E. coli. Okay? There are proteins that are out there that help to control transcription. Some of them turn off transcription, some of them turn on transcription, and we give them the group name of transcription factors. Transcription factors. So these are proteins that help to control transcription. Many of them bind directly to DNA. Bind directly to DNA. And somebody asked yesterday, well, how do they know which one to bind to? Well, they've got special structure that's in them that recognizes some very specific sequences in the DNA. That's how they do it. Okay? So you can imagine that transcription factors could bind at a promoter, for example, and help RNA polymerase to do something, and they can do that. And there are other ones that can bind at a promoter and actually stop RNA polymerase from acting. And we're going to see examples of both of those today. Okay? Well, there's a term I want to introduce, and they're called sequence elements. All right? And it sounds very big and hairy and so forth, but all a sequence element is a sequence that's recognized by a transcription factor. It's really all it is. It's a sequence in DNA that's recognized by a transcription factor. So the transcription factor is a protein, all right, and that's what a sequence element is. You see different names for different elements and so forth, and we're not going to spend a lot of time considering many of those at all. We're going to talk about a couple, basically, all right. So, but I want you to know what that term is, a sequence element. So a sequence element is something recognized and bound by a transcription factor. Okay. Well, one of the things with RNA that you sort of know is that we have a very long set of DNAs in our cells. Bacteria have not nearly as long as we do, but they still have a relatively long circular DNA that's there. The RNAs that are made from it, though, are much smaller. And they're much smaller because the RNAs have very specific places where they start and fairly specific places where they stop. And the idea is that the cell is making an RNA to make a protein, or in some cases, more than one protein. Okay? But the point is, it doesn't make an RNA that copies the entire genome. That's why specific start places and termination sites are really, really important. Okay? Well, one of the things we see when we look in the genomes of bacterial cells is something interesting. And the something interesting that we see is that related genes in bacteria are often grouped together. They're often grouped together. And this grouping together of the genes makes for a relatively simple way to control them. A relatively simple way to control them. I'll show you that in a minute, okay? But I want to give you a name first. There's a lot of names. We always name things. The name I want to give you is an operon. You see it right there. Okay? An operon is a name for a group of genes that are clustered together and they're all controlled by the same promoter. So it's a group of genes clustered together, and they're all controlled by the same promoter. Now this turns out to be simple, yet powerful for the cell. And we'll see how this comes up when I talk about the lac operon. Okay? I'll explain that to you. So everybody understand what an operon is? It's a cluster of genes that are all under the control of the same promoter. Okay. Now, the lac operon is a collection of genes. Okay? And the collection of genes, there are 3 of them in the operon. And this collection of genes is found in E. coli cells. Again, the most common organism we study, E. coli cells in our gut. Okay? And E. coli cells have this cluster of 3 genes called the lac operon. The 3 genes are essential for the bacterium to be able to break down the sugar lactose. Right? So the sugar lactose, it's also known as milk sugar. We'll see it later in the term. But for right now, you only need to know its name. It's just a sugar that occasionally bacteria encounter. Now notice I said they occasionally encounter. These bacteria are in your gut, right? You are not constantly drinking milk. I'm not. You're not. I suspect most of you are not. Some people hardly ever drink milk. All right? Well, those E. coli that are floating around in my gut, I don't drink much milk, for example. These E. coli floating around in my gut, if they are continually synthesizing the enzymes necessary to break down lactose, they're wasting energy. Because there's no lactose if I'm not drinking milk. Right? So cells need to have a way to turn on those genes when lactose is present and turn off those genes when lactose is not present. And so that's what I'm going to spend some time talking about today. Okay? All right. Now, the genes, there are 3 of them, all right? And for our purposes right now, it doesn't matter much what they are. We're going to focus more on the control of the synthesis of those genes. That is, the transcription of those genes. That's the first thing we're going to focus on, all right? All right. So, the 3 genes are located in a linear fashion, okay? And they would stretch to the right over here. When transcription of the lac operon occurs, it starts at the promoter region and it continues all the way through all three genes. So we get a messenger RNA that has all three genes on the same messenger RNA. Okay? Everybody with me? All right. Now, it's important, as I said, that the cell be able to control when those genes are being synthesized, when the transcription is going to occur, and then later, of course, when the translation to make them into protein will occur. Well, transcription is primarily the way in which this is controlled. Okay? Here is a schematic diagram of the promoter region. Okay? In the case of the lac operon, it's called the operator. The operator. Okay? The promoter region is called the operator. Okay? The operon has an operator. Oop. Don't lose that. Yes, indeed. Okay. Now, you'll see some regions that are marked up here. And, of course, when transcription starts, it starts here at the +1 and moves to the right. It turns out that there are two proteins besides the RNA polymerase that can bind in this region of the operator. Two proteins. And they pretty much control whether or not transcription will occur. Right? The first of them is called the lac repressor. Okay, so I'm going to keep this up here and I'll show you where things bind. All right? The lac repressor is a protein, and it's a protein that will bind over here on the right. It binds over here on the right. With the lac repressor, the name repressor suggests it's repressing something, and in fact, when the lac repressor binds right here, it does something very simple. It represses transcription of the lac operon. It stops the messenger RNA from being made. And the way it does it is very simple. It covers up part of the place where the RNA polymerase wants to bind. The RNA polymerase, therefore, can't bind. It's physically blocking the RNA polymerase from binding where it wants to, and consequently, no transcription occurs. Okay. There's another protein called CAP. It's called a couple of other things. It's called CAP, C-A-P. It's also called CRP. They call it CAP. We call it CAP here, and I like the word CAP. Okay. CAP is a protein that can bind way upstream. You see minus 87 to about minus 49. And when CAP binds, it actually helps the RNA polymerase to do transcription. So we have two proteins besides the RNA polymerase that are helping to control whether or not transcription occurs. Now, there's more detail to them I'm going to tell you in a minute, but I want you to understand what the proteins are before I move forward with that. Okay? So we've got CAP, and we've got the lac repressor, and we've got an RNA polymerase that's wanting to bind. Okay. All right, so this now shows the operon in a little different fashion. And you can see that the lac repressor can bind right in that region where I was talking about. Okay? The repressor binds right there. It physically blocks the RNA polymerase from getting in and doing its thing. Okay? That's a given. However, if it stays there forever, there will never be any proteins made. We want to have a situation where when there's lactose present, we have transcription, and when there's no lactose present, there's no transcription. And it turns out it's very simply controlled. The way it's controlled is the repressor is capable of binding a molecule called an inducer. An inducer. Right? The inducer, when the lac repressor binds to the inducer, it can no longer bind to the DNA, and it comes off. So the inducer is inducing transcription of the operon. The question then is, what is the nature of the inducer? What is the inducer? The inducer is actually a molecule that's made from lactose. To keep it simple in this class, we're going to call it lactose. Technically, it's a molecule made from lactose. We will treat it as if it's lactose. Okay? So, this is simple. Lactose appears in the cell because it's been floating around, and all of a sudden, the RNA polymerase, the lac repressor binds it. It can no longer bind to DNA, and the RNA polymerase can come in and it can start transcribing. That's all fine and dandy. That's wonderful. Okay? Well, what about CAP? How does CAP fit into there? CAP stands for cyclic AMP binding protein. You don't need to know that. Okay? Cyclic AMP binding protein, as its name suggests, binds to the molecule called cyclic AMP. And cyclic AMP, as we will see later, is a molecule that's involved in eukaryotic cells in signaling, telling the cell to do something. In prokaryotic cells, one of the things it can tell the cell is, hey, bud, I'm running out of energy. I'm running out of energy. CAP, when it binds to the lac repressor, when it binds to the lac operator, I'm getting my words messed up now, okay? What it does is, there it is, okay? When it binds to the cyclic AMP, that's the only time it binds to the operator, okay? So when the cell is really starving for energy and there's lactose present, transcription is turned on gangbusters. Because this guy helps the RNA polymerase to bind, and the RNA polymerase is not blocked, so it takes off and it starts synthesizing RNA like crazy. Okay? Well, what if there's no cyclic AMP? What if the cell is not starving, but there's lactose present? Will we have transcription? The answer is yes, we will. Or at least the E. coli cell will. Right? But it won't be as strong. Why won't it be as strong? Because the Pribno box of the lac promoter is not very good. It doesn't look very much like that TATAAT. It doesn't look as much like it. This helps to get the RNA polymerase there and says, start here, dummy. So the strongest transcription will occur when the cell is starving for energy, making cyclic AMP, and there's lactose present. When there's no lactose present, then the repressor will bind and nothing will happen, even if there's cyclic AMP. That's another one. What if we have no lactose and the cell is starving? Well, that's a time we definitely don't want to be starting transcription because we would totally be wasting energy if we did that. That would mean we'd have a lac repressor and we would have a CAP. But since the lac repressor is blocking the RNA polymerase can't bind, the lac repressor would win. That is, no transcription would occur. Yes, sir? I won't say it's, I think you're talking about the feedback inhibition, is what you're talking about. I wouldn't call it feedback inhibition, no. But I wouldn't parallel it to that, no, I would not. But the net effect, in a sense, I guess, is that it would, because if you think about it, you've got 3 or 4 genes that could be made, and you're turning all those off at once, then maybe in a sense it is. But I guess I don't think about it in those terms. But I can see why you would think that. Okay? But the beauty of this is all 3 genes are controlled by one promoter. One promoter is determining that all 3 of them are made or not made. Okay? That's pretty cool. It's very simple. Okay? When we look at eukaryotic cells, ooh, it gets really complicated. Okay? When we look at prokaryotic cells, we see the schemes are very, very simple that help to control whether or not transcription occurs. Okay? Now, the previous slide that I pulled up there briefly shows the genes that are actually made. Okay? Beta-galactosidase is one of the genes that it breaks down lactose. Okay? It's called the LAC-Z gene. The LAC-Y gene is an enzyme called a permease, and it helps to bring lactose into a cell. So you have to get a little bit in there first before all these things get started. That's true. All right? And the transacetylase is an enzyme that actually converts lactose into the inducer, but we don't need to worry about that. Okay? So the identity of the genes really isn't as important as much as what the actual control mechanisms are for determining transcription. Okay, questions about that? Yes, back there. I'm sorry? Okay, so that's called the permease. It helps to bring it in. So some lactose can get in without it, okay? But once the permease is there, then a lot of lactose will be brought into the cell. Do you have a question? No, I was back behind you. Sorry. I'm pointing the same direction for two of you. So the promoter is turning on the transcription that ultimately will code for the proteins. Okay? Okay, so the repressor will function as a tetramer. Yeah. Other questions? Okay. Well, that's a simple scheme. I'm going to show you a more complicated scheme that I hope you won't get too wound about. All right? But I want to tell you something about this because this is kind of a cool system. All right? So bacteria are, some people say, extremely simple, and other people say they're extremely complex because they have evolved to a point where they've gotten simplicity as a result of the evolution that they've gone through. It's an argument, it's a debate that goes on out there. All right? Well, when you see this one, you think, whoa, this is really a pretty cool system that these bacteria have evolved. It's called the regulation of the tryptophan operon. You hear it called TRP, TRP. The TRP operon is a much bigger operon than the lac operon. It involves about 10 genes. They're all under the control of the same promoter. And so the tryptophan operon gets its name from the fact that the genes in the operon are necessary for synthesizing the amino acid tryptophan. They're necessary for synthesizing the amino acid tryptophan. Now, when we think about the energy it takes to going into the synthesis of a messenger RNA for 10 genes, we're talking about a lot of energy. The cell wants to really make sure it's needing tryptophan before it starts synthesizing that. Otherwise, it's wasting a lot of energy. 10 genes, it has a lot of energy associated with making that. Okay? So the cell has a system set up where when tryptophan is absent, or in low quantities, it's never completely absent, but when the tryptophan concentration is low in the cell, the operon synthesis will be turned on. And when the tryptophan concentration in the cell is high, the synthesis of the operon will be turned off. Okay? Now, that's the setup here. The way in which it's controlled is a process called attenuation. And I'm not going to go through this whole thing with you because I find students get very worried about all the details of it. And I don't think that all the details are really critical. Right? The important thing is what the cell is trying to do. It's trying to make messenger RNA for the operon when the cell is needing it and not make it when it's not needing it. Okay? So I'm going to briefly go through and show you this, but I'm not going to go through in a lot of detail. Okay. It turns out that this operon is not controlled by starting the synthesis of the messenger RNA. That's what we saw with the lac operon. Either it started or it didn't. If the repressor is there, it doesn't start. And if the repressor is not there, then it can start. This one is not controlled at the level of whether or not it starts. It's controlled at the level of where does it stop. Where does it stop? Okay? So we saw in the lecture yesterday that termination is an important consideration, because termination stops the RNA polymerase from moving forward and synthesizing things, right? Okay. Well, that's part of what happens here. Part of what happens. This guy starts, the synthesis of the operon starts routinely. It's going on in the cell all the time. Okay? So the cell is always wasting a little bit of energy starting the synthesis. Okay? When the concentration of tryptophan in the cell is high, termination happens very quickly. Termination happens very quickly. On the other hand, when the concentration of tryptophan in the cell is low, termination doesn't happen until all 10 genes have been made. Okay? So when the concentration is high, a very short transcript is made that's basically thrown away. When the concentration of tryptophan is low, the entire 10 genes get made into one long messenger RNA. Once that messenger RNA is made, of course, the proteins can be made to start synthesizing tryptophan and increase the concentration. Now, all this is showing is that this little messenger RNA that the cell starts to make can have two different forms. That's all it's showing you. These can have different base paired regions, and if it forms this one on the left, it makes what's called a terminator. Remember the stem loop we saw yesterday? That looks just like that stem loop, and in fact it functions just like that stem loop. If this guy forms, poof, transcription stops. This will form when the tryptophan concentration is low. I'm sorry, the concentration is high. I'm saying it backwards now. This will form when the tryptophan concentration is high. All right? On the other hand, this form over here will form. It's called an anti-terminator. And it will form when the tryptophan concentration is low. This now says to the RNA polymerase, keep about your business and go ahead and continue synthesizing the messenger RNA through all 10 of those genes. This one says, stop right here. We've got plenty of tryptophan. Now, if you're curious about the mechanism by which each one of these forms, which is an absolutely fascinating thing, come see me. I'm not going to tell it to you here because everybody will get very worried about understanding it. But it's a really, really cool mechanism. Okay? So if you're curious, come see me. I'll be happy to explain it to you one-on-one. Yes? It is not a ribozyme activity, no. 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 So this, the activity actually has to do with it actually interfaces with the translational machinery. Yep. Okay. Questions? This is the first time I've spared the class this explanation. And this is one of the things, and the reason I'm doing that is because I found that so many students, A, were getting very upset with it, and B, they were spending an inordinate amount of time looking at this that might be one or two questions on the exam, and they weren't worrying about all the other bigger picture things. So I've given you the important components of this, and I'm not giving you the details about how each of these form. Okay. So the tryptophan operon is, and by the way, I should also say one more thing. The tryptophan operon is not alone in this scheme. E. coli have several operons for synthesizing amino acids that work exactly the same way. Very common mechanism for controlling the synthesis of amino acids in E. coli. A very common mechanism. Okay. Well, we've talked about prokaryotes, and I said eukaryotes is way more complicated. And the good news is I simplify there as well. All right? We're not going to go into all the detail because it would take 2 or 3 full courses to just begin to scratch the surface of eukaryotic transcription. We don't have the time for that, and I'm sure you don't have the desire for that. But I do want to point out some different things about eukaryotic transcription that are important to understand in general. Okay? E. coli, did you have a question? Yeah. So everything above here is prokaryote. That's right. Okay? And eukaryotes transcription is much more complicated, And it's much more complicated because, remember, we've got DNAs wrapped with proteins. We've got much larger DNAs. And in addition, in eukaryotes, genes are in pieces. They're in pieces. Those pieces have to be put together in eukaryotic cells. Prokaryotic cells, they don't have that. They'll have, every gene is intact. They may have 3 genes in an operon, but each gene will be intact in that operon. They won't come in pieces. It'll come in one, each one will come in one piece in that entire operon. In eukaryotic cells, virtually every gene is in pieces, and the pieces, meaning the messenger RNAs, have to be assembled after transcription has occurred. Okay? There's one level of complexity right there. Because of that complexity, eukaryotic cells do not, underline, do not have operons. Eukaryotic cells have all of one gene that starts out on one messenger RNA. Some of those pieces of the messenger RNA get cut out, and the other pieces get spliced together, but everything is synthesized for one messenger RNA contains one gene in eukaryotic cells. Okay. Another difference with prokaryotic cells is in prokaryotic cells, there's only one RNA polymerase. One. Okay? In eukaryotic cells, there are 3. The 3 RNA polymerases are called RNA polymerase 1, polymerase 2, polymerase 3. And the one we're going to focus on is polymerase 2. That's the one that makes the messenger RNAs. So RNA polymerase 2 is a eukaryotic RNA polymerase that makes the messenger RNAs. RNA polymerases are pretty complicated. Here is the RNA polymerase for a yeast, RNA polymerase 2, and look at the number of subunits. There are 12 subunits that are present in a eukaryotic RNA polymerase. No, we're not going to memorize those. But I'll tell you something interesting about RNA polymerases. And it's true to some extent also about DNA polymerases. The structure of them looks kind of like a hand grasping something. That's what it looks like. It's got a thumb and it's got fingers, and these fingers hold on to the DNA. The DNA slides right through here. That's the kind of basic structure that we see in the RNA polymerases and to a lesser extent in the DNA polymerases as well. Okay. Well, in eukaryotic cells, we have promoters. Okay? But eukaryotic cells, the promoters and the control sequences for transcription, which is what promoters are way more complicated than they are in prokaryotic cells. Why? Well, because the needs in eukaryotic cells for which proteins they need and how much they need are a lot more elaborate than what bacterial cells need. All right? We saw in the case of lactose for bacteria. Do I have lactose? Okay, I'm going to make the proteins. Do I not have lactose? Okay, I'm going to turn them off. In eukaryotic cells, we have much bigger decisions to make, like, hey, I'm a bone cell. I've got to make bone. Okay? I'm a skin cell. I've got to make, the things that skin cells need. I'm a liver cell. All right? So in eukaryotic organisms, we have specialization, we have differentiation of cells, and different cells have different needs. Now I want you to remember that all of the cells of the organism have the exact same DNA. You start out as a single fertilized egg, and your skin cells have the same DNA as your liver cells do. Why is a skin cell different from a liver cell? Because of the different pattern of expression of genes in a liver cell compared to a skin cell. It's the different expression of genes that give rise to the differentiation that we see that occurs in higher organisms. All right, so the promoters are much more complicated. But even with them, there are some common features that we can see. All right? For one, in the case of eukaryotic cells, we see something like the Pribno box. It's called the Tata box because it has a sequence instead of TATAAT, which is what the Pribno box has. In eukaryotic cells, it's basically TATAAA, and they call it the Tata box. Okay? The Tata box is located not exactly 10 nucleotides away from the start site. That's where we see it in the prokaryotic cells. But instead, in eukaryotic cells, it can be anywhere from about 25 to 100 bases ahead of the transcriptional start site. It's variable. It depends on individual genes. One gene will have it in one place. Another gene will have it in a different place. That's what I mean by variable. Okay? The Tata box is not present in front of every single eukaryotic gene. Ones that get made in large quantities usually will have a Tata box, but those that are made in lesser quantities don't always have a Tata box. Now, there are many sequence elements in eukaryotic cells like Tata box. I'm not going to go through those. There are dozens of them that are bound by specific transcription factors that help to control whether or not given RNAs are made. So I'm not going to go through all of the sequence elements. I'm not even going to really talk much about any of them, other than to point out that there are things called upstream sequence elements. These can be up to several hundred base pairs away from the transcriptional start site. Remember that eukaryotic DNAs are very big. They've got a lot of room to stretch out. Okay? In the prokaryotic cells, we saw the control elements being roughly 100 nucleotides away. These can be several hundred nucleotides away, and I'm going to show you some other elements in a little bit that can be several thousand nucleotides away. Okay? All right. Now, another difference we see in eukaryotic cells is in prokaryotic cells we saw sigma factor along with RNA polymerase bound to the DNA, bound to the promoter. Okay? In eukaryotic cells, the RNA polymerase doesn't bind directly to the promoter to start things. Other proteins come in first. Other transcription factors get the process started before RNA polymerase even gets there. Okay. The other proteins that come in are called transcription factors. In fact, they're called initiation factors. And we're not going to obsess with these either. Okay? I will point out one that I think you should know, and it's this guy right here, TF2D-TBP. TF2D is the first protein that will typically bind at the Tata box, because TBP stands for Tata binding protein. This guy helps to get the whole process started. And then after it binds, there's a sequential binding of these other proteins that are helping to make a transcriptional complex. That during that, as that complex is being assembled, RNA polymerase can come in and start transcription. Okay? It's sounding complicated. It is complicated. Right? So we're not going to go through the steps of that. But suffice it to say that there are a lot of proteins that are there to help RNA polymerase get the overall process started. Okay. If you want to see the process, there it is. I'm not going to hold you responsible for it. This shows you the steps in the binding. There's the TF2D with the Tata binding protein that is bound to the Tata box. We see other proteins coming in. There's TF2A, TF2B, TF2F, TF2H comes in, and TF2E, okay? And then finally, after all these things get going, the RNA polymerase, actually the RNA polymerase is in here, RNA polymerase gets started and synthesizes things. One of the things that has to happen in eukaryotic transcription, yes, you should know this, is that RNA polymerase must get phosphorylated. It has to get phosphates put onto it, and you see that happening right here, before the transcription will start. That's a sort of a start signal for the polymerase saying, okay, you're ready to go now. The RNA polymerase must get phosphorylated in order for the transcription to start. Now you'll notice the RNA polymerase takes off and does its thing. Yes? No, it's just in eukaryotes. Just in eukaryotes. Okay. You'll notice RNA polymerase goes off and does its thing, but TF2D can stay behind and start the whole process again for another RNA polymerase to come in and do its thing. Okay. The last thing I want to talk about is, with respect to eukaryotes, is an unusual set of transcription factors. They're called enhancers. And when people first discovered enhancers, they were pretty baffled by them. Remember that in a prokaryotic cell, all of the control sequences are only about 100 nucleotides away from the start site, and they're all ahead of the start site. All right? When they started discovering what enhancers were, they found some really bizarre things. They found, first of all, that enhancer sequence elements, enhancers are the proteins, by the way, the enhancer sequence elements are the things that the enhancers bind to. The enhancer sequence elements were located, in some cases, several thousand base pairs ahead of the transcriptional start site. And in some cases, they were several thousand base pairs down from the transcriptional start site. It didn't have to be before it. It could be after it. Well, that kind of flew in the face of everything that they understood about how transcription worked in prokaryotes, because in prokaryotes, those sequence elements are close to the RNA polymerase can bind and open things up and get polymerase going. So how can something be downstream from the start site and function? Or how can something be thousands of base pairs ahead of the start site and tell the RNA polymerase where to start? Okay? Well, enhancers work like this. Okay? They actually don't have to be close. In fact, here is an RNA polymerase, here's a promoter, and here, thousands of base pairs away, is an enhancer sequence element with enhancers bound to it. Look what the DNA has done. It has bent backwards to hold on to this and help transcription get started. So enhancers are transcription factors that bind to enhancer sequence elements and interact with the RNA polymerase by a folding back mechanism, kind of like what you see here. This is a very nice diagram. I just found it recently. Okay? Very beautiful diagram showing what's happening. As a result of this, these enhancers can affect transcription of genes that are hundreds or thousands of base pairs away. Okay? Now, we don't talk about it here, and so I'm not going to hold you responsible for what I'm going to tell you, but I'll tell you briefly what one of the reasons that these enhancers are important so far away. They're involved, excuse me, in helping to clear out those histone proteins. Remember, we've got histone proteins that have got to be cleared out. One of the things that they are involved in helping to do is clearing out those histone proteins. You're not responsible for that, but that's a very cool thing that the enhancers are involved in doing. Okay. Here's some different response elements. These are the sequence names for the sequence elements that are found in eukaryotic cells. No, you don't need to know these. But some of these do very specific things. And one of the things that enhancers do that I think that you should know is that enhancers help to give tissue specificity. They help to give tissue specificity. How do they do that? Well, let's imagine that here is an enhancer sequence element. Let's say glucocorticoids. All right? So this sequence element, when it is present, Okay? If an enhancer is present, it will bind to it and it will stimulate the synthesis of a glucocorticoid, the glucocorticoids themselves. Okay? Well, not all tissues of our body make glucocorticoids, and not all tissues of our body make the enhancer that binds to it. Only the cells of our body that make glucocorticoids have a glucocorticoid enhancer. So now we begin to see how it is that a liver cell can make one set of proteins. A skin cell can make a different set of proteins. Some may be in common, okay? And some may be might not be used by either one, but instead would be used by a blood cell. Yes? These are not epigenetic. No. Epigenetic is a different phenomenon. Yep. Okay? Okay, so enhancers give tissue specificity. That's one of the reasons that we see enhancers in eukaryotic cells. We don't see them in prokaryotic cells because prokaryotic cells don't have tissues. Okay, that's a lot of stuff. A lot of stuff.